Hello. As Christina mentioned, I'm Daniel Saptari. I live in Lexington, Massachusetts, and I was diagnosed with osteosarcoma in my left femur during the summer of 2020. It caused me to miss out on a lot of the sports that I enjoyed, including tennis, running, biking, and skiing. I then underwent 9 months of MAP chemotherapy, and I'm now in remission. And the surgical side of my treatment was a lot more eventful. I went through 15 months of surgeries, none of which fully succeeded in giving me the function I wanted in my leg. So now I'm awaiting a rotationplasty surgery, which I hope will give me the function to enjoy the sports that I used to enjoy. On the brighter side, I now enjoy building and flying model planes, competing in my school of science Olympiad team, studying cellular biology and human anatomy and physiology, and other various science-related projects. As you all probably know, the whole process of osteosarcoma, it can be overwhelming and the future is uncertain for the patient, but it's largely because of people like Dr. Jason Eustein that I could grasp at least some of what was going to happen in my future. Dr. Eustein is a pediatric oncologist and physician scientist at Emory University and Children's Hospital of Atlanta. With significant training in molecular and cellular biology, Dr. Eustein has been fortunate enough to design and unify his laboratory research and clinical interests. He recognizes the need for improving our understanding of the molecular pathogenesis of sarcomas and identification of new therapeutic avenues, especially for patients with therapeutic resistance and/or metastatic disease. His laboratory has tremendous interest and experience in merging innovative murine models and patient-derived resources towards garnering molecular insights into sarcoma initiation, development and resistance, and metastatic progression. And eventually translating these findings towards testing novel therapeutic interventions for these aggressive malignancies. Thank you, Dr. Eustein, for all the work you do, and thank you all for being here.
Thank you, Daniel, for that kind introduction. Thank you, MIB, for this opportunity to chair this session. I'm really excited about chairing this preclinical models session. We have 4 outstanding speakers, and of course we're going to try to continue to uphold the outstanding session that already occurred before with the biomarkers. That was truly, as Kurt was saying, Dr. Weiss was saying earlier, an outstanding session that was presented. So we're hopefully going to keep the trend going, and we're excited about it. And as I mentioned, we have 4 speakers that come from all over the country, which is really exciting. I'll briefly introduce each of them. So I have Dr. Ryan Roberts, who's a pediatric oncologist, physician scientist, associate professor at Children's Nationwide. And he's also a member of the Translational Therapeutics program there. Ryan has been doing an outstanding job of really investigating metastatic disease in his laboratory, and also has been one of the key members of this proxy consortium, which we'll talk about much more in a few minutes. Very exciting initiative that was spearheaded by Philemon de la Cruz at Sloan Kettering, but also obviously Ryan at Nationwide. And Ryan also is the COG Biology Committee Chair for osteosarcoma, and he's been doing that since 2020. So his talk will focus on decentralized multi-institutional clinical trials in mice, the proxy consortium. Then our next speaker is Dr. Anand Patel, who's also a physician scientist. He's at St. Jude's. His research interests focus on combining genomics, patient-derived experimental models, and computational biology. You've already heard some of the computational biology approaches before from the previous session, so Anand's going to add to that as well. And his focus has been on development of some 3D models of osteosarcoma and towards mapping and modeling heterogeneity in pediatric osteosarcoma. So that'll be the second talk. The third talk is by Dr. Fan Yang, who's at Stanford University. She's an associate professor there, and it's really exciting. I'm really excited to hear about her work in terms of she's within the orthopedic surgery and bioengineering program there at Stanford, and she's also co-director of the Stanford NIH Biotechnology Training Program. She's really on the cutting edge of developing some hydrogels and unique micro and nanoscale properties to promote stem cell differentiation, tumor regeneration, and immune modulation, and using these approaches to study disease biology, including musculoskeletal diseases such as osteosarcoma. So she's going to present some of the insights into her 3D models. And our final speaker, last but not least, is Dr. Lori Graves, who's from Duke University. She is an advanced sarcoma research fellow at Duke. Her focus is on therapeutic resistance or escape, and the title of her talk is going to be Cross-Species Osteosarcoma Models towards intratumoral heterogeneity as mechanisms of resistance to chemotherapy. So it's my pleasure so first off, again, when it comes to questions, they're all open to as difficult or as easy questions as you want to give them, all right? I'm not like Troy in terms of setting up one over the other, so they're all equal. So equal opportunity for everybody here. So again, my pleasure to start it off. It'll be Dr. Ryan Roberts.
Thank you all. It's a pleasure to be here and to be here representing really a group of investigators. This is not work that I have done personally, but it's a work that we've done as a group, and really, as mentioned, a lot of the thanks should go out to Phil De La Cruz, who was unable to be here, but who has been kind of a leader in this effort. But I want to talk to you a little bit about kind of the main problem, one of the main problems that we have in studying rare cancers, and osteosarcoma being a great example of that, and that is that we individually, none of us see a ton of these patients. While several of us work at busy academic centers and tertiary referral centers where we get lots of patients, that lots of patients really pales in comparison to say, breast cancer or something like that, where there's just people coming in the door every day. And so when we go to study these diseases, trying to find the right models for different kinds of questions can be kind of challenging. And so to address that, a group of us from several different centers across the country, you can see them there on the map, got together and said, I think we should kind of consolidate all of the models that we've been building, and you'll see over and over the use of this word PDX, which stands for patient-derived xenograft. And what these are is when a patient is diagnosed and has a biopsy or has a lung tumor resected, we can take a piece of that tissue and we can propagate that tissue within a mouse, and the mouse becomes kind of an avatar of each individual patient. And you can see that once we put all of our resources together, we have many different PDX models available of many different types of pediatric cancer, but that includes like 147 at the time that this was made, different patient avatars of osteosarcoma. And once we start to have those kinds of numbers, we can start thinking about doing things that we couldn't really do otherwise. And so the aim of this proxy consortium is that we're bringing together multiple investigators, each of whom is developing their own experimental PDX models, and we're trying to enable both basic and translational research that can then be used to make decisions about go, no-go decisions for different therapeutics, that can make different types of questions that would otherwise be unanswerable available to the different members of the consortium, and to facilitate the sharing of our different models by showing what everyone has and how we might combine those in ways to answer different questions. And so the model that we've been working on for multi-center preclinical investigation takes the traditional PDX model, and this is still an experiment, so note that we're still kind of working through how we can do this. But what we're saying is that we can actually do clinical trials in mice, right? We can do multi-center, multi-institutional clinical trials without any of the regulatory barriers and without any of the expenses that we incur when we have lots of patients in a clinic. And we can just do it with our mice and our little mouse hotels and our vivariums in a multi-center approach that increases rigor, that shows that we can do this across multiple different models with multiple different genetic signatures, or maybe a single genetic signature, as I'll show you in a minute. And we can show that everyone gets the same result or doesn't, right? Just like happens in a normal clinical trial. And we can standardize the way that we do things, and we can help. And all of this, what this does is that this increases both, not just feasibility, but also the rigor and the reproducibility of the conclusions that we reach when we distribute the experiments across multiple different centers. And so the way that this might work is that someone might bring an idea to the group to say, I really think that this drug might be interesting in osteosarcoma, right? Why don't we test it? And so then we'll have a discussion as a group, and we'll talk about how this drug might best be utilized and whether there's a signature that should or shouldn't respond to the drug. We'll come up with a plan for how we think this should be tested. We'll present that plan sometimes to a company that might be the company that has the drug, and sometimes they'll give us the drug, and that then is distributed from the organizing center at Memorial Sloan Kettering across all of the different institutions with a plan for how the drug should be reconstituted, how the drug should be administered, what kind of mice we should be using in the trial, and then each individual center will enroll their mice in this clinical trial, right, and follow the protocol just like we would in a clinical trial, and then return the results to a centralized repository in a standardized format so that it all can be combined and analyzed. And we can do lots of different kinds of studies this way, and we can blind and unblind in different ways to mimic what might be done in a human clinical trial. And then this can be done, again, at multiple institutions, each of whom also has their own referral centers and can increase our reproducibility. Oh, this slide. Sorry about that. So as an example, we've been working on a project to look at different ways that we might target MYC-amplified osteosarcoma. This has been kind of a little bit of a hot topic, that we know that osteosarcomas that have MYC amplification tend to be more aggressive, tend to be more metastatic. And so we can say, well, what happens if we target some of those kind of super enhancer mechanics, specifically within those tumors that harbor MYC amplifications? And so we've gone across the consortium, we've decided what constitutes a MYC amplification for this study, we've written up procedures, we've actually gotten some companies to donate drug, the drug has been distributed, and these trials are now ongoing, and we hope to have the results soon. We can also consolidate all of the different models that we have, and so we're in the process of depositing all of our data into a federated database held at the organizing center at Memorial Sloan Kettering, and they have built a system that will allow us all to kind of sift through all of the different models available from all of our different colleagues, so that we can figure out what kind of studies we can do, and so that we can plan around and we can request the right models and tissues, both based on patient characteristics and on genetic genomic characteristics, to make targeted and precision medicine also part of our repertoire. And so we're going to see how this works. It's kind of an experiment in progress. It's been fun to work with all of these different investigators. I will say there's been a huge amount of work done by many of the staff, and especially the legal teams, to make this possible. We now have master agreements and template agreements in place at all of the different institutions, both for data sharing and for resource sharing of the models and of the agents. And a special thanks to Andrika, who has now moved on to a different group, but we did need a professional cat herder to make this happen. And to Phil, who really drove a lot of the process development and has been kind of the champion for all of this behind the scenes. And so we're going to see how this goes. We think that it's a cool model and a way to maybe accelerate some of the things that we were talking about earlier. And happy to answer any questions after the panel's done.
Great. So let me start by saying thank you to all of you and to the organizers for inviting me. My name is Anna Patel. I'm a pediatric oncologist from St. Jude, and I'm representing a group of, several of us who have been working together to try to develop models of pediatric solid tumors. And today I'm going to tell you about some of the things that we're thinking about moving forward. So this is going to be a high-level talk. And is going to focus on, how we are trying to think about bringing some additional models that may help speed up and help us scale some of the studies we'd like to do in osteosarcoma moving forward. So a major challenge for treating pediatric cancers in general, and cancers in general, are the heterogeneity that underlie these tumors. So in addition to the non-malignant components within the tumor, and those can include things like blood vessels, the extracellular matrix, the fibroblasts, as well as the immune system, we've also learned that not every tumor cell, every malignant cell within the tumor is created equal. And so there are varying states within these compartments. And so maintaining that heterogeneity as we bring these models into the laboratory is really paramount, because at the end of the day, we want to be as close to the patient scenario as we possibly can. So what I hope you guys take away from my talk today is that there's no one-size-fits-all for experimental models. Each model has its strengths and limitations. So for the last 10 years at St. Jude, particularly led by Beth Stewart's group, we've been collecting and generating orthotopic patient-derived xenografts by taking patient samples, purifying the cells, and injecting them into mice. And we're proud to be part of and participating in the Proxy Collaborative for that. But there's still limitations to the xenografts, even though we think that they're probably the closest we can get to the actual patient tumor that we started with. And the chief issue being that they take a long time to generate, and so we're limited by the amount of scale that we can achieve with a xenograft system. It would be very powerful if we could start to move towards some of these culture-based in vitro models, which would allow us to move much faster. And so I'm going to tell you about some of our efforts as we're starting to think about and starting to move some of these xenograft models into what we call organoids, which are 3D models, 3D cultured groupings of cells, and also starting to generate cell culture models. And I'm also going to tell you about our efforts to benchmark to understand how well we're doing in preserving that original patient heterogeneity as we move through these different models. So this has really been spearheaded by Dr. Justina McAvoy, who has led our 3D, 2D cell factory group. And the idea here is that her and her team take xenografts generated from our childhood solid tumor network portfolio and purify the cells and plate them onto ultra-low attachment plates. And what this does is it forces the cell to form into clusters of groupings that we call spheroids or organoids. And these look down here in the bottom. And you can see these sort of white circles, which are actually thousands and thousands of cells that are clumped together to basically form a heterogeneous mixture of cells. We then take these organoids and plop them onto coated plastic dishes and allow those cells to spread out and colonize the plate. And apologies, I think my movie isn't working, but what you would be seeing would be that sphere, those cells kind of spread out to form all of sorry, it's not forming. So those cells will actually spread out to form a single layer of 2D cultured cells. And along every step of the way, we have been very careful to try to preserve and make sure that what we're actually keeping and generating is actually really coming from the tumor itself. And so multiple stages along the way, cells are taken out, DNA is purified, and then we make sure that they still match up with the original patient sample. And then importantly, after 3 passages, we also take these cells and put them back into a mouse and make sure that they can still generate xenografts. So far to date, we've taken about 150 samples through this process, through a variety of different sarcoma and pediatric cancer types. Our major subtype has been rhabdomyosarcoma, which has kind of been our proof of concept tumor. But now we have about 14 osteosarcomas and another 15 that are underway. In total, we've had about a 70% success rate for generating lines from these models. And we plan to be able to post and distribute these through the Childhood Solid Tumor Network alongside the xenografts, which were sort of the originating tissue for this tumor. So importantly, we need to understand what we've gained and what we've lost from every stage in this process. And so alongside this, we've also been really focused in making sure that we can generate data to understand the heterogeneity within each of these model systems. And so we've taken 35 of these patient tumors that were osteosarcomas. Some were from metastatic sites and some were from primary bone sites. We've also taken orthotopic xenografts as well as spheroids, and we've begun to start profiling them kind of holistically to understand what's the difference, what changes as we take these samples through the process. And so that includes bulk sequencing, which we perform using whole exome sequencing, whole genome sequencing, and RNA sequencing. We also have been doing methylation profiling using bead arrays. In the thinking that we think that methylation can be a powerful classifier for sarcoma moving forward. And so far, we've also been very active in generating single nucleus RNA sequencing. And so we've seen a little bit of single cell sequencing data already in the previous session. The benefit of single nucleus RNA sequencing is it allows us to use bank tissue that has been frozen in our bank at St. Jude. We purify the nuclei and then take it through the same analysis pipeline. We're also starting to think about proteomics, and then finally spatial profiling using two instruments that have recently been installed at St. Jude. One, the Akoya phenocycler that gives us protein heterogeneity, and then finally the Cosmix SMI, which gives us, at the RNA level, heterogeneity. So I'll close by giving you some very, very preliminary borderline reckless data, which really is fresh off the sequencer. So, so this is the first 27 tumors that we profiled using single nucleus RNA sequencing. 10, importantly, came from metastatic lesions, 9 of which were from the lung, and 17 from bony sites. In total, it accounts for about 226,000 single nucleus profiles. And so we have a lot of work, but I'm looking forward to really digging into this data to now start to clean up this data, understand how the tumor microenvironment changes between and by comparing both the primary and metastatic sites, and then start to generate analogous data sets from the orthotopic xenografts, spheroids, and the 2D cultures. So I'll close by saying that this is always a team effort. I want to single out Mike Dyer, who really has been kind of the lead, and he's my mentor, as well as Justina McAvoy, who has led the 3D, 2D cell factory, and then Beth Stewart, who has really been the engine for our xenografting efforts at St. Jude. And then, of course, this would not have been possible without our patients and their families who have been very generous in both donating tissue, but also being the motivating factor behind all the work we do. So thank you, and I'll move to the next. Oh, and sorry, I always forget this slide. And for researchers that are interested, any of the xenografts are available through the Childhood Solid Tumor Network, and as we get closer, I don't have a timeline yet for the cell lines and the spheroids, they will also be available there. Thank you.
All right, I think Ananda actually did a great job of introducing what I'm going to talk about more. And before I get into my research talk, I want to thank the organizers for actually inviting me here. And also I want to particularly thank Tyler's family. I don't know if Tyler's parents are here, for also partially supporting me to attend this conference. I think I am probably one of the very few engineers in this room. I'm not a doctor. And I don't have an MD. In fact, my background is in bioengineering. And my lab at Stanford has spent a lot of time developing biomaterials to regenerate human tissues, including cartilage and bone. And a few years ago, we actually were intrigued by, why don't we use all these engineering tools that we have initially developed for stem cell differentiation and regenerating bone, but for studying why certain cancer arise from bone, and how does the bone niche actually change, their signaling and their drug response. So that's the story that I want to tell you today. So as I mentioned, you already heard from the previous speaker that bone is actually containing multiple cell types. But one thing I also want to highlight is that bone also has very unique matrix. So it is actually a tissue in our body that is highly mineralized, right? If you think about majority of the tissues in our body, it's actually, it doesn't have minerals. But bone, actually, if you look at a matrix, 70% is actually minerals, and the rest of it is organic proteins, and collagen 1 is one of the most abundant one. And also, you have heard a great intro from the last speaker about the experiment models for cancer research, and also the pro and cons. And the 3D models that I want to talk about today is a little bit different than the spheroid model or the organoid model you have just heard about. And the difference here is actually introducing not only just the cells, right? Because the tissues is actually made of a lot of cells plus what we call extracellular matrix. So the kind of 3D models my lab is very interested in developing is to actually incorporate those extracellular matrix using bioengineering approaches, but also importantly, try to make it scalable so we can actually make this in a relatively cost-effective manner, but also being able to do drug screening with much, much shorter timescale compared to animal models. So previously for osteosarcoma in particular, actually I would say this is a really understudied area. If you look at the field of using tissue engineering for modeling cancer, there has been tons of work done for breast cancer, for brain cancer. But when I was starting this work a few years back and I was looking into the literature, there's very little work actually on applying engineering approaches to study osteosarcoma. There's some work on using spheroids, but the limitation is that there's really no extracellular matrix cues. And also there's some emerging work using some hydrogels or collagen plus collagen sponge plus hydroxyapatite scaffold, but those works are very, very preliminary. They use only a few very well-established immortalized cell lines, and also the outcome analysis is really limited, and there's no in vivo validation. So we identified this as an opportunity for us to contribute to the osteosarcoma community by really designing scaffold with bone ECM mimicking signals, and also really integrate the model with multiple PDX cell lines. And for the outcome analysis, we collaborate with also clinician scientists and also biostatistician to do comprehensive signaling analysis. And finally, using the model, if we can identify any drugs, our pipeline is to then validate that using animal models. So before we started work, we actually first asked Alex Lee, who is a biostatistician in our collaborators, Alejandro Sweet-Cordero's lab at UCF, to do just a data analysis to compare osteosarcoma versus all the other solid tumors. So what you can appreciate here is the pathways that are enriched in osteosarcoma. A lot of this are actually related to collagen and bone minerals. So this really strengthened our hypothesis, but also rationale of designing scaffolds with these cues to study osteosarcoma. And the question we're asking is, how does this bone ECM cues impact osteosarcoma signaling? And also, what can we learn from this about informing drug screening? So to do this, we went back to the tools that my lab has already developed for bone tissue engineering, where my lab has patented this very unique gelatin microribbon hydrogels. So you can imagine them to be basically fettuccine-shaped hydrogels that are like building blocks, and we can mix this with cells. You can imagine the cells to be like the little meatballs. So you can put them together. They can actually cross-link into a three-dimensional tissue. So this is our three-dimensional osteosarcoma model. And then we look at various outcome analysis, and finally look at also in vivo validation. So as I mentioned, when we started this project, actually, I started this project without knowing my collaborator. So we were using a few immortalized cell lines. But once we established the baseline, actually I reached out to Alejandro Sweet-Cordero, referred by one of my colleagues at Stanford's orthopedic surgery. And we were very happy to obtain multiple PDX cell lines that his lab has established, but also very well characterized in terms of their genomic feature and using various sequencing technique. So we then combine our model with those cells, and then here you can actually appreciate the morphology of those structures. So these are, as I mentioned, they look kind of like fettuccine shape, and we can coat them with or without hydroxyapatite, which is mimicking the bone minerals. And one thing you can instantly appreciate here is that once you put this tumor into 3D model, their proliferation is much, much slower compared to 2D, and it mimics better the in vivo proliferation kinetics. And then we looked at also the drug response, because as we know, a lot of drugs that people screen using 2D study actually fail to work in the mouse model. So basically there's a fake drug sensitivity. And what we saw here is that when you translate, when you actually culture these cells in 3D, the drug resistance actually increased substantially. And also when they see mineral cues in the bone, the drug resistance further increased. So this motivated us to then look at more signaling, and we started with IGF-1R because this is one that has been actually elucidated by one of the previous paper showing that this is 3D culture is really critical in retaining this signaling in Ewing sarcoma, but its role in osteosarcoma, the effect is unknown. So we then looked at this using our model, and you can see again using various techniques that the signal is only actually expressed when we grow these cells in 3D, and it's further enhanced when you actually have the mineral cues. And this is very interesting that we also see that if we block the signaling, you basically can see that this directly modulates the drug resistance as well. So importantly, because in our model we saw that HA further enhanced the drug resistance, so that actually made us think that, okay, does that mean that if we go in vivo validation, it's really critical to validate this model using actually orthotopic bone tumor rather than just do sub-Q model, right? Because a lot of people also use sub-Q because it's faster and easier. So that's actually indeed what we saw, suggesting that our in vitro model actually is a very useful way for us to also understand the critical role of bone mineral in osteosarcoma drug resistance. And later on, we also went into looking at protein. So this is actually some selected data sets from just 2 PDX cell lines. And here you can see from the heat map that in 2D, actually in 2 of the cell lines that we have tested, the RTK phosphorylation signal is mostly lost. What does that mean? That means that if you're screening using 2D culture for drugs that are targeting RTK, you're going to see no response. But that's not because the drug may not work. It's because the model basically no longer retains those targets. So it really highlights the importance of using appropriate 3D models for testing drugs that targeting RTK in particular. And also, if you look at RTK, if you look at RNA sequencing, here are some examples of a couple of cell lines. And again, we validate that our 3D model, and especially the one with mineral cues, cluster the best with the patient tumor as well as the mouse models. So with this, we have now started to really look at the genes that are upregulated, specifically in 3D and patient tumors, but are lost in 2D models. So this would help us to be able to identify basically 3D specific drug targets that would otherwise be missed out. And here I just want to show one example that, using the sequencing data set we have, we basically identify 2 potential drugs, and then we use our 3D model to first do the testing. And as you can see here in the first row, actually, if we look at the 2D culture or just 3D without minerals, we actually see very minimal drug response. But when we actually culture these cells in the mineral-containing 3D models, we see a much stronger drug response. And this was then validated using the mouse models as well. But using the 3D model, it took us a few weeks to do the study. But using the mouse model, it took us 6 months to validate the same study. So I think this really tells us the value of having 3D model that has physiological relevance to speed up the drug discovery process. So what are we thinking about going forward is that here we're looking at things in a very simplified way, right? We're doing bulk sequencing, and we have heard from a lot of speakers earlier that this is a very complex disease. So we're very interested to do also integrate single cell sequencing in the future to think about understanding genomic complexity. And also use this for not only targeted therapy, but also for combination therapy screening as well. And also, as you have seen from my slide earlier, the drug resistance is much higher in 3D, similar to in vivo, but lost in 2D. So this also offers us a great tool to study drug resistance as well. And we can easily add additional bone niche cues as needed to understand the mechanism or the contribution of other bone niche cues in osteosarcoma. And in the end, patients actually suffer from metastasis. That's really the key challenge. So in tissue engineering field in my lab, we have also developed spatially patent tissues to allow us to actually study metastasis from one tissue to the other. So that's also another direction we're very interested in moving forward. So with that, I want to thank Eva, who is a recent graduate PhD alumni in my lab, who really started all of this work, and our collaborator, Alejandro Suet-Colero, and his team from UCSF, as well as our funding sources. Thank you.
Good morning. My name is Laurie Graves. I'm a pediatric hematology and oncology fellow from Duke University, and I'm thrilled to be here at Factor. I want to say thank you to the organizers and to the family of Anna Lee for the support in this presentation and for being here today. My presentation today will be on some ongoing unpublished work that our lab has explored pervasive intertumoral heterogeneity as a mechanism of evolutionary escape from chemotherapy. So this audience is keenly aware of the challenges that the profound tumoral heterogeneity and chemoresistance that is found in both primary and recurrent osteosarcoma tumors plays in limiting effective treatments for some patients with osteosarcoma. However, less is understood about how changes in these heterogeneous populations evolve over time in response to and in release from chemotherapy, particularly as it relates to minimally viable populations that evolve and then go on to develop recurrent chemo-resistant tumors. So our objective to address this question is to characterize and ultimately be able to intervene upon these evolutionary dynamics that occur within heterogeneous tumors that contribute to the growth of chemo-resistant relapsed tumors. So a lot of this work began as we explored the response to chemotherapy in patient-derived organoid models. And I appreciate the prior speakers for introducing 3D and organoid models to you today. Our work has obtained tumor biopsies both from canine and from human patients with osteosarcoma, both at the time of diagnosis or recurrence or metastasis. And from that, we've digested these samples and plated them into 3D matrix to generate organoids. And from those organoids, we're able to perform drug screening, genomic sequencing, or tumor or organoid banking. And as an example, organoids generated from 11 different canine osteosarcoma patients are displayed here on the x-axis. And what you can see is that after treatment with doxorubicin, we're able to demonstrate significant intertumoral heterogeneity in response to chemotherapy, with tumor organoids on the far left demonstrating greater sensitivity to chemotherapy versus those on the far right demonstrating significant resistance to treatment with doxorubicin. And to take this further, we also observe intratumoral heterogeneity using this 3D organoid model. By taking each tumor section as it arrives to our lab and dividing it into smaller, spatially distinct subsections, we're able to generate organoids on a greater resolution capacity. And as we demonstrate here, 6 pieces from this one particular tumor, D090, these organoids demonstrated a range of responses to chemotherapy, ranging again from quite sensitive on the left to quite resistant on the right. And we're able to use this model to really then isolate those chemo-resistant organoid subpopulations and perform RNA sequencing on these organoids to hopefully identify both pathways that are upregulated in these resistant, more aggressive organoid populations, as well as potentially targetable pathways as well. So the heterogeneity that we have observed in our organoid model prompted us to question whether there could be greater heterogeneity that evolves over time in response to chemotherapy. And so to explore this question, we turned back to our cell line models and have adapted these to chemotherapy to generate chemo-resistant models. These models are adapted to chemotherapy with escalating doses over time and re-exposed to drug to demonstrate resistance relative to their drug-naive counterparts. And displayed here are 143B human osteosarcoma cells, well-established human cell line, as well as 17-3X, a patient-derived cell line that we use in our lab. I apologize that some of these are a little bit off. So to further dive into our the clonal dynamics over time in response to and upon removal of chemotherapy, we utilize a CRISPR-based barcoding system to be able to lineage trace these populations over time with the development of resistance and upon release from chemotherapy. So in this release model, I'm just going to click to get everything on the screen. In this release model, again, we adapt these cells to escalating doses of drug to generate the resistant population. And then we remove drug from culture, and we collect cell populations at multiple different time points in this period of chemo release at 1 week, 2 week, 4 week, 8 week, 12 week, and 6 months after release from chemotherapy. And in the top 2 graphs on the right, we demonstrate that the orange population, which is the 4-week released population, upon re-exposure to chemotherapy, continues to display profound resistance, really similar to that of the cells that are chronically adapted to drug. So even after release from drug, removal from culture, these cells are continuing to demonstrate chemoresistance. And as we performed RNA sequencing on these resistant and released cell lines at different time points, we demonstrate that there's preservation in gene expression of well-known drivers of chemoresistance, including ABC transporters and CYP enzymes. But what's interesting about these released cell lines is that not only do they display chemoresistance and preservation of expression of doxorubicin resistance drivers, they seem to regain a proliferative advantage over the chemo-resistant population, demonstrating greater ability to form colonies and greater rates of proliferation over time. So in summary, what we're seeing upon this chemo-released population is preservation in resistance, but with a greater growth advantage, not dissimilar to what we see in patients at the time of relapse. So to delve back into the clonal dynamics in these populations over time, we turn back to our lineage tracing barcoding system. And we see heterogeneity in our naive population, as demonstrated by a number of different alleles present in the far left. Upon administration of chemotherapy and adapted to resistance, our resistant cells in the second from the left bar demonstrate having undergone a profound selective sweep, but some preserved heterogeneity with some smaller diverse clones at the top of the bar. And ultimately, what we see over time with release is that there isn't an evolution in our clonal dynamics, with ultimate expansion in some of those smaller diverse clones with time. And at the 4-week population, in which we were seeing greater proliferation and a growth advantage, we're seeing new clones, new predominant clones, I should say, emerge and proliferate. And so next we wanted to compare this heterogeneity that we're seeing in our clonal dynamics to phenotypic heterogeneity and growth patterns. And so to do this, we took single cells and ultimately grew each population from single cell clones up through logarithmic growth through confluence. And what we see is we do see that preserved heterogeneity kind of in phenotypic growth patterns in the green naive cells on the far left. But we do see some reduction in that heterogeneity and growth patterns in the resistant cells on the right. Though notably, there is still some preserved heterogeneity and growth, with some growing more quickly and some single colonies only ultimately taking up to 100 days in culture to reach logarithmic growth phase. And as we incorporate the growth dynamics of our released cells in orange, we see that they overlay some of the faster clones of the resistant population, suggesting that these released cells may arise from a subset of the heterogeneous population of the resistant population. And ultimately, as we perform RNA sequencing on these populations over time to further understand some of the changes in clonal dynamics with chemo release, we do see upregulation in several different pathways that correlates with the emergence of an aggressive phenotype in our 4-week release population, including epithelial to mesenchymal transition, angiogenesis, and other cell cycle and inflammatory and cytokine release pathways. So ultimately, in conclusion, despite the selective pressure of chemotherapy, heterogeneity is maintained across our chemo-resistant models. Yet upon chemo release, these highly proliferative clones with preserved chemoresistance and a distinct gene expression pattern emerge from these populations, yet still maintain heterogeneity, modeling recurrence in our osteosarcoma patients. Ongoing and future work will aim to further explore these mechanisms of chemo release and proliferative growth advantage in additional preclinical models, including our osteosarcoma organoids, in which we're able to demonstrate heterogeneity even at a single organoid level, and in the pulmonary microenvironment using ex vivo pulmonary metastasis assays. Ultimately with a goal to be able to therapeutically target these chemo-release cells prior to proliferative outgrowth. And I'd like to thank my mentorship in the lab, Dr. Ewart and Dr. Samarelli, clinically, Dr. Wagner, as well as multiple other lab and veterinary partners, and our funding support as well. Thank you.
All right, just want to thank all 4 speakers for outstanding talks, and I guess we'll open up the floor for questions. I see there's one question for Anand. Are the osteosarcoma cells implanted into a bony site in terms of femur or tibia?
Yeah, that's, we are strong believers in the orthotopic model, so I was delighted to see Dr. Yang's data. So yeah, we usually inject those paratibially or into the femur.
Hi, I have a question, for Drs. Roberts and Patel. So this wonderful collection of PDXs, eventually people will have to use them in mice, but you know that primarily mice should be skid mice, and they are not very suitable, or I would say not suitable at all for testing the biologics, such as antibodies. The pharmacokinetics in those models are completely different of the charts. And how do you see the future of testing like biologics in PDXs? Will it be all in spheroids? Will, say, FDA or other regulatory agencies accept that spheroidal data versus mice? Where you see it's all going? Thank you.
So that's a really good question and observation, and I think that when we, there's different kinds of biologics, and we can usually get around PK problems if the problem is just how long does the antibody last in the mice and things like that. But for instance, if the agent is, just as an example, an antibody drug conjugate that's targeting something that's on the tumor cells, we can get decent efficacy data from the PDXs, although we don't get good toxicity data necessarily from the PDXs, but that's not different from any mouse study. We also there are other cases where the modeling can be done really well. So Jason didn't get to talk about his stuff, but there's some really good ways that you can model some of those things in immunocompetent mice using some of those models. And then also, like with some of our veterinary colleagues that are in the audience, a lot of those, when done correctly and when validated the right way, some of those questions can be tested there. Ultimately, the best model is the patient, right? And so we won't really know on any of these things until we get there, but we can prioritize and we can work through some of the problems that we might have when we do rigorous preclinical testing and hopefully increase the probability that they'll succeed.
Maybe I'll add one comment here, is that I think one actually potential benefit of using the 3D tissue engineered 3D model is that you can actually selectively introduce population of cells of interest. And one thing that actually one of the PhD students in my lab is working on is actually to look at also how tumor-associated macrophage actually interact with osteosarcoma. And also, the big picture, one thing we are curious also is that, osteosarcoma does not respond very well to cancer immunotherapy, which works very well for many other tumors, right? But we don't really understand why. So, if we can understand the mechanism that they kind of evade, right? But you need to study then the tumor immune cell crosstalk, which is hard to do when you're using an immunodeficient mice.
I will say though that they tend to be actually pretty good models for cellular therapies. And so a lot of cellular therapies are pretty easy to test in the SCID models, but all of these are complementary and answer different questions in different contexts and scenarios.
Yeah.
So a quick question allied to that. There are immunodeficient mice with a humanized immune system. Do you have experience of implantation into those, such as the Mr. G?
My experience is that I hope to work with them as little possible. No, I mean, yes, you can do those things. They are not
very reliable.
simple. They're not reliable. They take very long periods of time. They're incredibly expensive. They're difficult but possible.
Yeah, I can basically vouch for everything Ryan just said. They're a very powerful system, but they're very fragile and require a tremendous amount of effort to keep alive and to process, to work with.
In the right setting, sometimes that's the right thing to use. And when that's the right thing to use, we should do it. But it's not simple, and it takes a lot of time and money.
Reproducibility. Yeah, good.
John Healy, New York. Spectacular presentation. Thank you. It's essential to the drug discovery process and higher throughput, and I compliment you on that. I want to know, how close does it have to be to the normal physiologic situation to enable you to go to the next level? It's somewhat an extension of the previous question. But, you know, for example, it's not just collagen. I mean, how many of the matrix proteins do you need to have in the so-called osteoclastogenic profile with fibronectins and things in order to promote metastatic profile? Next, other physiologic aspects. For example, vascularity, pressure, pH, oxygen tension. You know, what oxygen tension do you grow things under? And it drastically alters the chemosensitivity. And then back to the perennial question of, well, are you looking at primary tumors or metastatic ones? Well, these chemophysiologic parameters become very important once you get to be a centimeter, right? And that's what is going to kill a patient. The micro metastatic disease, if it stays micro, it doesn't kill anybody. And it's non-definable from your radiologist. Indeterminate nodules, right? We suffer from that dilemma all the time. But when you get to a centimeter, all of these issues of vascularity, perfusion, drug delivery, large molecules don't penetrate by diffusion. At that point, it's all by convection. And so the engineering part of this and how it interfaces is essential. And you don't have time to go into all of those things, but just your perception of it. And back to the original question, how close does it have to be in order to allow you to go to the next stage?
All right, maybe I'll try to answer that first to share some of our perspectives. In fact, I think it's never possible to mimic everything using in vitro systems or using mouse systems as patients, right? And for what we're doing, actually our goal is to determine the minimal components that is necessary. Because otherwise the model is too complicated to scale, right? Because you have to test hundreds of combinations. If it takes like so much time to just make one, it's not going to be scalable. So I have another PhD student in my lab, and her project is to compare all different scaffolds. So we're trying to understand what is the key thing that is really necessary for the signaling to be retained that correlate with the drug response. And I also think that while we cannot capture all the signaling, if we could capture signaling that would otherwise be missed out using existing models, that would still be valuable, right? There are certain signaling that you may be needing like 10 different cell types or the oxygen tension or all the stuff to be present, to be identified. We may not be able to identify some of those, but if we could identify some of the other ones that are, you know, require, say, 3 components to actually identify that, and shows that, because in vivo validation and also eventually patient validation is necessary, right? But this would at least give us the throughput to, I would say, to accelerate discovery process. And for your second point, I totally agree. A lot of the challenges actually are very different when you scale up. And this is where I think engineering actually has moved a long way. Like, for example, in my lab, as I mentioned, a lot of our project is actually working on tissue and organ, you know, engineering. You know, I have collaborated with other people using, you know, the same material I was talking about here, but 3D bioprinting, so you can actually print them into very large scale. And you can actually introduce tissue-engineered blood vessels. You can actually introduce potentially shear forces if needed. We just haven't used that for osteosarcoma yet because we're not quite there yet. But my feeling is that those challenges are actually lower. Like if we're ready for testing some of those, like whether it's integrating with, tissue scale, oxygen tension, those are the things we have already developed for all the tissue engineering stuff that we can easily repurpose for studying osteosarcoma.
John, the most honest answer to that is that if we knew what would predict a response in clinical trials, we would all be using the same system. But we don't, and so we do our best. However, I think some big picture historical lessons are worth discussing, and that is I'm not aware of an agent in any cancer that's been wildly successful in the clinic that's not also wildly successful in almost every model that you tested in. And the opposite is not necessarily true. Not every agent that's been wildly successful in preclinical models has been wildly successful in the clinic. So I do think that it's a screening tool. What I do think, this is my soapbox, so I'm just going to say it, but I think we've got to get away from getting excited about modest tumor growth delay, because that's not exciting, that's progressive disease, and that definitely does not predict success in clinic. So we've got to be looking for things that are wildly successful in our preclinical models, and those should be prioritized to test in the clinic.
Maybe time for one more question? Good.
Hi, thank you for a great panel. In the work that you presented on the clonal evolution through treatment and emerging of resistant cells, so the expression in those resistant cells, does it overlap or match the expression that is seen in the poor prognosis clusters that were presented in the previous panel?
It's a really great question. I would have to see that just in remembering from some of the previous slides, I do recall some overlap, particularly like with inflammatory signaling, cell cycle progression pathways, et cetera. But would love to explore that in more detail with some of the prior panel speakers.
Thank you.
All right, well, lunch is next. I want to again thank our tremendous panelists here for their great presentations. And if you have any follow-up questions, feel free to come up and ask them, and enjoy your lunch. Thanks, everybody.